Hello everyone. InformationBoxTicket Lifestyles brings you today molecular biology topic on real-time PCR. We will learn about its definition, theory, types, steps and application. The temperature-dependent nucleic acid amplification method, known as polymerase chain reaction, is used to enzymatically amplify DNA or RNA in vitro. It was created by Kelly Mullis and his collaborator in the middle of 1980s and is one of the most powerful and crucial tools in molecular biology and genetics today. The principles of nucleic acid replication and hybridization are combined in this process. This non-culture based nucleic acid amplification method allows us to quickly create billions of copies of a single DNA or RNA segment. Since its creation, PCR has undergone a number of changes leading to the availability of many PCR procedures for various uses. The two PCR kinds that are most often employed are reverse transcriptase PCR and quantitative PCR (qPCR). Reverse transcriptase polymerase chain reaction or RTPCR is a specific kind of PCR method that in vitro amplifies RNA enzymatically. It is the only kind of PCR that has the capacity to amplify RNA. The reverse transcriptase enzyme is used in addition to the other fundamental PCR building blocks. First, reverse transcription, which is mediated by reverse transcriptase enzyme, converts the sample RNA to complementary DNA, cDNA. The PCR procedures uses these cDNA molecules as a template for amplification. mRNA or microRNA analysis and gene expression research are done using RT-PCR. So let's see the table of content. We will learn the objective of RT-PCR, principle of RT-PCR, its requirement which is enzymes, the type of RT-PCR, the steps of RT-PCR, applications, advantages, and lastly limitations. Before starting this video, don't forget to like this video, subscribe to my channel and press the bell icon button. Let's start with the objectives of RT-PCR. Its objective is to make billions of copies of a single RNA segment by amplifying the particular RNA segment, and to identify certain illnesses, genes, and to investigate gene expression. The principle of RT-PCR. The reverse transcription and standard PCR processes are combined in RT-PCR. The reverse transcriptase enzyme initially converts the sample RNA to double-stranded DNA, complementary DNA, during the reverse transcription process. The two single-stranded DNA templates can then be created from cDNA by heat decomposition. Based on nucleic acid hybridization principle, primers can anneal to their complementary sequences in these single-stranded DNA templates. Then, using the DNA replication principle, DNA polymerase lengthens the primer by successively adding the nucleotides to the 3' end to create a double-stranded DNA. Denaturization, annealing, and elongation are three procedures that are cycled through repeatedly in order to produce millions of copies of cDNA while controlling the temperature of the reaction. Let's see the requirements, enzymes of RT-PCR. Number 1 comes the nucleic acid sample, which is the sample RNA. RT-PCR uses RNA as its sample as opposed to other PCR methods that use DNA. The sample used most frequently is messenger RNA. Prior to amplifying, the RNA will be transformed into cDNA. Then comes the reverse transcriptase enzyme. It is an enzyme that causes the RNA strand to split into two complementary DNA, cDNA strands. It produces central dogma reverse and goes by the name of RNA-dependent DNA polymerase enzyme. It is a crucial part of RT-PCR because it turns sample RNA into cDNA for amplifying. Number 3. DNA polymerase enzyme. By assembling the nucleotides sequentially in accordance with the template strain, DNA polymerases are enzymes that catalyze the synthesis of complementary DNA strands. The most extensively used DNA polymerase is Taq DNA polymerase, which was isolated from bacterium Thermus aquaticus. Taq DNA polymerase is thermally stable and keeps working even after repeated cycles of heating and cooling. Number 4. Primers. In RT-PCR, three main kinds of primers are utilized. Number 1. Random primers. Number 2. Oligo-DT primers. And number 3. Sequence-specific primers. Number 1. Random primers. For the purpose of cDNA synthesis using reverse transcriptase, these are the brief single-stranded sequences of 6 to 8 nucleotides that bind at the complementary side of RNA with or without poly A. Number 2. Oligo-DT primers. These are the repeating deoxythymidine DT containing oligonucleotides, typically 12 to 8 nucleotides long, that bind to the poly A tail of messenger RNA. Number 3. Sequence-specific primers. These are the brief single-stranded nucleotide sequences that bind to the particular area of sample RNA that is of interest. Most often, it is used in one-step RT-PCR. Number 5. Deoxynucleotide triphosphates DNTPs are synthesized nucleotides that are used in cDNA synthesis and the creation of additional strands of cDNA during amplification. Deoxyadenosine triphosphates DATP, deoxyguanosine triphosphate DGTP, deoxythymidine triphosphate DTTP, and deoxycytidine triphosphates are the 4 distinct DNTPs that are employed. Number 6. PCR buffers and other chemicals. And lastly, thermocycler PCR machine. Kindly don't forget to subscribe to my channel. Types of RT-PCR. RT-PCR may be divided into two forms depending on whether the reverse transcription and the amplification stages take place in one reaction or tube or two separate reaction or tubes. There are two types of RT-PCR. One is one-step RT-PCR and the other is two-step RT-PCR. Let's dive into the one-step RT-PCR. It is a kind of RT-PCR in which the amplification and reverse transcription processes takes place in the same tube. One tube is used to combine all the necessary ingredients. Reverse transcription first takes place, creating cDNA, which is later amplified by PCR. Advantages of one-step RT-PCR over two-step RT-PCR. Number 1. It features an easy setup and handling. Number 2. It is more precise and accurate. Number 3. It is less likely to be contaminated. And number 4. It is quicker and less expensive way. Disadvantages of one-step RT-PCR over two-step RT-PCR are: due to the use of many chemicals in a single reaction tube, it detects fewer templates per reaction combination. Number 2, it needs a bigger template to start since the template detection is less accurate. Number 3, it prohibits the storage and subsequent examination of cDNA produced during the reaction. Number 4, primer, dimer, and non-specific binding have a higher likelihood. Number 5, reaction failure is somewhat common occurrence. Then comes the two-step RT-PCR. It is a different kind of RT-PCR in which reverse transcription and amplification take place in two different tubes. Reverse transcription takes place in the first tube producing cDNA. The PCR mixture is then introduced to another tube with the cDNA there, where they are amplified. Advantages of two-step RT-PCR over one-step RT-PCR are: number 1, we can save cDNA produced by reverse transcription. Number 2, greater efficiency, precision, and dependability are also present, and more templates may be detected per reaction mixture. Number 3, reduced likelihood of primer-dimer bonding, non-specific binding, and reaction failure. Disadvantages of two-step RT-PCR over one-step RT-PCR are: number 1, the likelihood of contamination is increased. Number 2, it is a more difficult and laborious procedure that needs more resources and a skilled individual. Steps or procedures of RT-PCR. Reverse transcription and amplification are the two steps that make up the fundamental process. The process differs from one-step to two-step RT-PCR as well. However, the fundamental procedures are the same for both types and may be broken down into four stages. Number 1, the preparatory stage. Number 2, reverse transcription stage. Number 3, amplification stage. And lastly, the product analysis stage. Number 1, preparatory stage. RNA extraction and the preparation of the whole reaction mixture are done at this initial stage. Prior to extracting the sample or bringing it from storage, all materials must be organized, safety precautions followed, and PCR reaction preparation environment sanitized and all reagents brought to working temperature. In one-step RT-PCR, primer, DNA polymerase, DNA polyadenylase, DNTPs, buffers, reverse transcriptase enzyme, RNase H, and all other components are added in a predetermined and calculated amount to a single reaction tube. Following that, the tube is placed into a thermocycler for further processing. In the two-step RT-PCR, reverse transcriptase, RNase H, primers, DNTPs, and other buffers and chemicals are added into a tube along with the sample RNA. The tube is then placed into a thermocycler and heated to a certain temperature where cDNA are produced. Number 2, reverse transcription. The RNA is first transformed into cDNA in this stage, which is followed by amplification. In a tube, the entire reaction mixture, including reverse transcriptase, RNase H, DNTPs mixture, primers, nuclear-free water, reverse transcription buffer, and other components in one-step RT-PCR is added. The tube is then heated in a thermocycler to a temperature of 40 to 50 degrees Celsius for 10 to 30 minutes to complete the amplification process. The primer will attach to the appropriate RNA sample site at this temperature, and the reverse transcriptase enzyme will create cDNA by adding the free DNTPs. Number 3, amplification. This phase is comparable to the DNA amplification process used in other PCR procedures. The same reaction mixture is put through an amplification procedure in a one-step RT-PCR. The cDNA is extracted and put into another tube where DNA polymerase, primers, PCR buffer, DNTPs, and other reagents are introduced into the two-step RT-PCR at the same time. After that, a thermocycler is used to amplify the signal using the tube. Denaturization, annealing, and elongation all take place in succession during the amplification process for a predetermined number of cycles. Number 4, product analysis stage. The reaction mixture exposed to PCR is examined in the following step to determine if the intended amplification was accomplished. For the product analysis, the gel electrophoresis technique is frequently employed. This extra step is not required for real-time RT-PCR. Don't forget to subscribe to the channel. Applications of RT-PCR. Number 1. Study gene expression. For the traditional Norton blot approach to evaluate and study the gene expression, a bigger messenger RNA sample is needed. However, by amplifying the little messenger RNA sample and examining the nucleotide sequence by RT-PCR, we can examine the gene expression. It is employed in researching and locating genes that are expressed in pathogens that are multi-drug resistant. Number 2. Identification of unknown species. HIV, SARS, dengue, HCV, and other viruses may all be detected with RT-PCR. In order to identify them, numerous bacteria and even higher species, ribosomal RNA and messenger RNA, are also investigated. Number 3. Infectious disease diagnosis. The RT-PCR technology is used in clinical laboratories to diagnose various viral infections, bacterial, fungal, and parasitic infections, cancer cell disorders, and genetic illnesses. Number 4. Gene insertion and gene therapy study. Eukaryotic messenger RNA, which has no introns, may be introduced into prokaryotes and is converted into cDNA with the help of RT-PCR. Gene insertion and gene therapy outcomes are monitored using RT-PCR. These processes are intended to translate various kinds of messenger RNA sequence, which in turn exhibit distinct gene expression and codes for a specific protein. The RT-PCR method may be used to examine this particular messenger RNA sequence. Number 5. Study mutation and cancer cells. Tissue-specific mutant alleles can be identified and measured using RT-PCR. It may not spot any unwanted alteration to the messenger RNA sequence and certain messenger RNAs that are exclusively made by various cancer cell types in our body. Number 6. Tools of genetic engineering and viral study. In genetic engineering, RT-PCR is used to analyze changed DNAs and their transcribed RNAs as well as to amplify target RNA. Let's see the advantages of RT-PCR. Enzymatically, millions of copies of messenger RNA may be produced using this quick process of RNA amplification. It is quite easy to use. A thermocycler controls and operates the process semi-automatically without the need for human intervention. It is affordable yet has a very high specificity and sensitivity. It is a fairly precise approach for determining whether or not an individual has exposed to RNA virus. RNA viruses can be divided into strain-level classifications. It has reduced the time needed to detect viral infections and RNA viruses. Compared to the conventional Northern blot method, it can only identify a very little quantity of messenger RNA, around 5 pg. Gene expression and mutated genes may be quickly and readily investigated. This has made it feasible to research gene insertion, identify cancer in its early stages, and track the effectiveness of gene therapy. It may be used to both identify and measure the sample RNA because it is qualitative and quantitative procedure. Limitations of RT-PCR. Only RNAs, especially messenger RNAs, may be amplified by it. For the purpose of creating primers, prior knowledge of RNA sequence is necessary. Because the system depends on enzymes and operate at full temperature, even a little variation in reaction temperature will reduce the enzyme's effectiveness. Therefore, a stringent system of temperature regulation is needed. A little amount of contamination with a comparable primer binding site can be magnified and produce an incorrect result, either positive or negative. A tiny quantity of organic or inorganic contamination in the reaction mixture can have a significant impact on the reaction. It takes a complicated reaction mixture and a trained individual to run the procedure, which is quite laborious. So that's it for today. Thank you so much for watching till the end. Don't forget to like this video, subscribe to my channel and press the bell icon button so you don't miss any of my upcoming lectures. Thank you.